So we're going to start with the textbook with the cell cycle notes. So if you want to do a split screen with your textbook, you can. Our notes look like this. We're using chapter 8, page 5 through 10. And we're going to just do a little bit of this together, and then I'm going to leave the rest up to you. So if you scan this, there are 15 questions from the textbook, and then we're going to get to the game. So I'm going to go over here to our textbook so that we can see it up on the screen as large as possible. If you go to page 5 in the textbook, there's an awesome video. I meant to play this for you right off the top, but I'm going to do that here when we go over, after we go over a few things. So when you're in the textbook, and you know what? I highly encourage you to be in the textbook, even if we're not going through it. This is how you get an A in the class. I don't know if I did this to your class or not. You're the first class in the topic, so sometimes I add things as I go. And I want to remind you to not distract yourself with your iPad on the internet. Don't go to shopping. Don't play games on the internet. What you can do is you can daydream, and you can be in our textbook, and you can look around during class. You know, that's what an A student does. They don't just stay on task and wait for the teacher's timing all the time. Question?
Oh, yeah. I was just wondering if this is going to be the game online.
Yeah, it is. We're going to go to the library, and you're playing it on your iPad. So we'll get to that. So back to my point here is an A student gets a lot done during class. They almost get all their homework done during class. Even the nutrition, 15, 20 minutes. Some students might be doing their homework so that they don't have any when they get home. It kind of whatever works for you. But those that have a really busy schedule, those that are highly involved in sports and choir and performing arts, they're usually A students, and they have very little time for homework. And they do most of it during class. So if you're catching yourself being distracted on your iPad because you want to watch this video, you know, I can walk around and see people watching soccer videos, football videos, videos on YouTube for TikTok, and they're, you know, watching like Netflix, and, you know, you can distract yourself all you want, but is that going to really help you reach your goals? So there's a time and a place for entertaining yourself, and it's not during class. So if you're not an A student, check to see how often you are distracting yourself. It's fine to distract yourself with the textbook. So when I'm on a page, are you highlighting what you see I have highlighted? Or are you highlighting extra things on your own? Or would you open up the textbook on your own at home? If your parents ask you, do you have any homework? And all you think is, worksheets? No, I got my worksheet done. Nope, I don't have any homework. But if you're not preparing yourself for quizzes on like a daily basis, or certainly a weekly basis, and all you're doing is cramming at the end, you're really good at getting A's on homework, and then you get C's and D's on tests. So you have to own the process of making sure you master it however you do. There's too many tools for me to assign it all because it would be too much, but you have to figure out what works for you. There's flashcards over here. We're in this chapter. You should know all the key terms. We're going through it kind of step by step, but you should make sure you're drill and practicing. Would you do flashcards like when you come into class and then just zip through them? If you did that 5 times in 2 weeks, you're going to be a master at this material. And then come test time, the night before the test, you're probably not even going to study. You've got it down already. So if you want help with this, if you want help getting from your C or D average on tests up to an A, then it is something that you should learn quicker than later because it can serve you right now instead of learning it the hard way. All right. On the last exercise, you had to identify cells in different phases of mitosis. Do you know what happens in prophase? You know what happens in metaphase? So there's a textbook chapter 8, page 5, that goes over all of that. We're not going to spend too much time with this, but you do need to know it. So this is up to you. I'll just quickly go over it. And I know it, but if I just speak it, it might go over your head, so you should practice this. What do you call the DNA when you can't recognize chromosomes? Is it sister chromatids? Is it chromosomes? Is it chromatin? See, these are 3 words that we went over. And if you didn't spend some time to try to master it, like that Etch-a-Sketcher, it just takes a little bit of time and then you get it. It's really easy after a certain point. It's just a learning, and then you're at that plateau and then you learn much quicker once you have the method that works for you. It's labeled right there. Obviously, it's chromatin. When they get shorter, it's called chromosomes. So interphase is where a cell spends 90% of its time. It's just growing. You might realize, hey, I need to copy my DNA, but you still can't recognize chromosomes during interphase. The only time you start to see chromosomes is when it enters the M phase. Letter M stands for mitosis. Mitosis is the division of the nucleus. So it has to get ready to separate all the copies of DNA. So when a cell divides, both cells have a copy of the DNA. It's simple enough, right? So in prophase, there's no order or fashion to where the chromosomes are. They're just haphazardly wherever they started to form from chromatin. Other things are happening also. There's these fibers right here which are going to pull the chromosomes apart. They're called the spindle fibers. And they're just starting to form. So this is happening in G2, at the end of G2, where the spindle fibers are forming. And now we'll go over here to metaphase. Metaphase, the chromosomes line up in the middle. It's called the equatorial plate. This is a globe, 3-dimensional circle. So the chromosomes are lined up anywhere. Imagine another floor in the middle of this room. All the chromosomes would be lined up on that plate. Could be over there, could be over here, but they're all like in the middle of the room. That's called the metaphase plate. The spindle fibers are attached to these guys, and that's how they kind of push and pull them so that they're all in the middle. These are called sister chromatids to each other. It looks like 2 chromosomes, like 2 fingers together, but that's 1 chromosome with its copy, and there's a spindle fiber attached to both copies. So now when the director in the Hollywood movie goes action, A for action, A for anaphase, the actors are moving on the stage. And the sister chromatids get pulled apart, going to opposite ends, but as they're moving, that's anaphase. When they get to their destination over here on the right, this is telophase. And now all the chromosomes have been separated, and then they're going to start to unravel and then become chromatin again, where you can't recognize individual chromosomes. Mitosis is prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. You want to know an easy way to remember this? Anybody have a dog that you're training? During the pandemic, a lot of people got dogs. You have a new dog? Was it hard to potty train him? Did you have a certain method of doing that?
He was pretty good.
Yeah? There's like 4 methods. One is you crate them, called crate training. You keep them in a small space. They don't mind it as much as we think because dogs are pack animals and they're burrowing as well. So they don't mind, they feel real safe. So you keep them in there until they're potty trained, and then you take them out. When you take them out, you don't let them walk. You pick them up and you take them outside, and they have to go to the bathroom, so they go outside. You do that for a week or 2 weeks and they're potty trained. The other way, which isn't so good, takes a lot longer, and it helps them realize, hey, I can pee in the house. You put a mat down over in the corner. You can buy them at the store. They're pee mats. And you just pick up your dog and you put them on that mat, or they're supposed to know they can pee in the house on that mat and only in that spot. And then you got to train them to not pee on the mat and just pee outside. So skip the mat. Don't let your dog pee on the mat. Pee mat. P for prophase, M for metaphase, A for anaphase, and T for telophase. So right now, do a quick check for yourself. Where are you? Are you on the internet? Are you surfing because I was telling a story and you didn't have to write something down? So are you a student that is just looking to fill in a blank? So did you write down on your paper PMAT somewhere in the corner? And then did you write P for prophase, M for metaphase, A for anaphase, T for telophase? So you should get to a point where you go, hey, that's going to help me remember that. That's a good thing. So notes are not just, see that on the board, copy it down. See that, copy it down. See that, copy it down. That's a bad way of taking notes. How about if you were to go through the textbook yourself? Would you take notes yourself? I'll count that as extra credit toward the end of the semester. So if you want to take notes on your own and then turn them in periodically and at the end of the semester, and you go, hey, look, I just learned how to take notes on my own. Wow, that's awesome. You are going to go far because you are owning the process of what works best for you. Some people are going to make their own flashcards. Some people are going to make their own Socrative Quizlets. So you have to go the little extra mile, and it will make you an expert, just like that person with the Etch-a-Sketch. Try to be the master. We made a slight adjustment to your grades. So I sent that out to you in the last half an hour, if you noticed that. So some of you that I had a progress report, maybe your grade went up. But you know what that did? It moved the top score in this class was, I think, 101%. There are others that are at 99.5%. So if you found yourself down in the 70s or 80s, there was a lot of room for people that outcompeted you. If you are on a cross-country race, they left you behind. They're already miles ahead. So it's time to catch up with your performance if you are struggling to get a solid grade. It's not hard. This class is the easiest class that you'll have. It just takes a little bit of you organizing yourself so that you own your education. You're not just waiting for a teacher to give you a worksheet and then you fill in the blanks. I make my own worksheets. I did this, I think I did it last night. Took me 20 minutes or 30 minutes. I went through the text information on what I wanted you to get out of it. But it wouldn't take you very long once you get the habit of doing this on your own. And when you do, you're really at a college prep level. So some of you are still a couple of years behind. We recognize that during the pandemic, some haven't had a lot of academic practice. And so you're in 9th grade, but maybe you're at a 7th or 8th grade level with the way that you are academically preparing yourself. So we want to close that gap. If you have an A on homework and a CD on tests, it shouldn't be more than 1 grade lower on tests. Tests are a little harder than homework. You should never turn in homework that is not your best quality work because you totally own that process. So you can easily get an A in any class if you have an A on homework, an A on projects, and a C on tests, you'll get an A. But as soon as you start letting your homework and projects go from a B down to a C, then you're going to have a hard time getting a C in a class. All right, let's get to the worksheet. What are the stages of the cell cycle in order? It's okay if you were answering that question while I was talking and doing something academic, but if you were waiting for me to show it to you, then try to catch yourself. Where was I? Was I being distracted by my iPad? So that is section 4 where we left off. You sketch this. So in order, it is interphase and then PMAT. Interphase and then mitosis, prophase, metaphase, anaphase, telophase. Interphase is broken down into 3 stages, right? G1, S, and G2. So you could say interphase, G1, S, G2, mitosis, prophase, metaphase, anaphase, telophase, and that separated the nucleus. And then you have to separate the cytoplasm, cytokinesis. So once again, interphase, broken down into G1, S, and G2, and then mitosis, prophase, metaphase, anaphase, telophase, and then cytokinesis. It's all in that circle, starting right here. Interphase G1, interphase S, interphase G2, mitosis, PMAT, prophase, metaphase, anaphase, telophase, and then cytokinesis. So at these different color barriers, there's checkpoints. The cell is going to control itself with chemicals. Sometimes the chemicals are outside of the cell, sometimes they're inside the cell. Certain things have to happen in order for these chemicals to be released, and then it's going to start some more dominoes. Then there's a gap in the dominoes, and then there has to be another push to get it started again. So that was question number 1. Number 2, how does cytokinesis, dividing the cytoplasm, differ if you are a plant or an animal? Hey, before I turn the page, I'm at the bottom of page 4, and you see this? It has a video on a tutor. This isn't a video. I think this one is an audio. So it's a good idea for you to listen to those, especially if things seem fuzzy for you. Question 2, cytokinesis is dividing the cytoplasm. So after the nucleus divides, there's 2 nuclei in one cell. This is an animal cell. Animal cells are round, and it just pinches off. It's called a cleavage furrow, and it will keep pinching like a string tied and tightening, and then it will pinch it all off. So 2 cells are the result of that cleavage. Here's a plant. Plant cells have a hard outer covering. It's the cell wall. The cell wall is made out of cellulose, which is a type of sugar. It's like hard candy. So there's no way that the cytoplasm could even be pinched because the outside is hard. So in plants, a new cell wall starts in the middle, a little bit here and there is a patchwork. It continues to form from the middle on out to the sides. When it finishes, it has 2 smaller cells with the same outer shape that it had originally. That's called a cell plate. So the answer for the question 2, animal cells will cleave or indent to the point where they get separated into 2. But plants are going to form a new cell wall starting in the middle of the cell toward the outside. It's called a cell plate. It's labeled right there. Just another 5 minutes or so, and then we're going to get to the game. We're now going to talk about how the cell controls itself. we've all had a cut before. After it stops bleeding, you notice, wow, there's a big gap between my cells. They used to be touching and now they're not. That is what is referred to as anchorage dependence and density dependence. So look at this experiment that they had. This is kind of like a cut where cells get separated from each other. And that's going to start them dividing. And they'll divide until they start touching each other again. So that's density dependent. When cells have formed a complete layer and start touching each other, they stop growing. They control that. That control comes from the outside. And then anchor is like an anchor on a boat. It's going to stop the movement. So the cells aren't going to grow if they're kind of floating, but as soon as they get anchored in place, that's going to trigger them into growing. We've talked about cancer. See this last one down here? It says that cancer is when your own cells lose the ability to control when they should grow and when they should stop dividing. And so cancerous cells are cells that have lost the ability to control their cell cycle, and they keep growing and growing and growing, and they become a clump of overlapping cells, and then you can start to feel that. If it's near the outside of your body, you can feel the lump. If it's on the inside, you might not know it until it starts to push against other cells and nerves, and now you're starting to feel pain. So listen to your body. You should know what normal feels like, and if it's abnormal, then you should keep an eye on it and try to address things sooner rather than later. Yes? No, we're going to be breaking pretty soon, so if you can wait 5 minutes. Otherwise, if you can't, go ahead and take a break right now. I'm going to do one more question with you, question 5. Some chemicals outside of the cells can turn on cell growth, and here it is. If you are with the textbook, it is page 7, chapter 8. Chemical factors can also influence the rate of cell growth, and it is called growth factors. They're chemicals that are secreted and they stimulate other cells to grow. Human growth hormone, for example, is a chemical that your body puts out, and if it's present, your cells are encouraged to grow. If you don't have this chemical, then the cells are going to stop growing. So there's a chemical that is going to start the G1 phase and the S phase. There are dozens of different types of chemicals. In general, they're called growth factors. There are some that are going to turn on new blood vessel formation. But I'm not going to quiz you on the names of the different chemicals and what they are. But we're just generally going to know that there are chemicals that are going to turn on the cell cycle so that it can go through a checkpoint. So there's a checkpoint at the beginning of G1, checkpoint at S, a checkpoint at G2, a checkpoint at mitosis, and there's even a checkpoint between every phase of mitosis, between prophase and metaphase and anaphase. So it's all controlled by chemical signals. And we can finish question 6 because it's right here at the bottom of page 7. I highlighted nearly the entire paragraph right here. Cancer cells are different than your normal cells because they can no longer regulate their own cell cycle. They're not subject to anchorage dependence. They're not going to worry about density dependent. When they hit each other, they're just going to keep growing, whereas they're supposed to stop growing. So something went haywire in that cell. These bad cells should destroy themselves, or your own immune system should destroy them, but they can trick the checkers into thinking that they're normal, so they just keep growing out of control. So it's up to you to answer the rest 7 through 15 for your homework. I'm going to explain the game, and then we're going to head over to the library's rotunda so that we can have a new environment to work in. So to play the cell game, you're going to click on this. As I mentioned, it's from the Nobel Prize website. And you can run through the game without worrying about writing anything down. I played the game like 5 or 6 different times. And I'm like, what's a good takeaway? There's no scorecard, but you're going to answer some questions throughout the game. And you are presented with 4 scenarios. So I have 4 pictures from the game. So notice right here in the game, it has the cell cycle. G1, S, G2, and mitosis. So when it got to this point at G1, it's a little hard to read on my printout. The cell cycle growth has started. Cell has high energy. The cell is increased its size by 20% right here. What's next? It says, circle a highlighter, put an arrow next to the correct answer choice on the computer screen on the left-hand side. So what should happen next? Should it check the cell size or should it check the genetic material? This game is a little bit trial and error. You'll have to realize by understanding the cell cycle, what should happen right here at growth 1? Growth 1 is growing. So the checkpoint should be, have I grown large enough? So you would hit check its cell size. If that's the correct answer, it will produce a new message in this screen, and then it might give you more options. See these buttons right here? You're going to hit those buttons as answer choices. So there's nothing wrong with you making the wrong choice. If you do, this guy is going to go, wrong choice, try again. You have 10 seconds. If you do another wrong one, he goes, uh-oh. And then the thing will like explode. So you are this guy right here. You are the control center inside of the cell cycle, and you have to choose the right things in order for the cell cycle to keep going. So if you're hitting the wrong buttons, you're having the cell cycle stop and the cell isn't going to be able to proceed. So you're going to be presented with information about the cell cycle. You are this guy right here. It is called like the stimulus response and the cell killer. So at times the cells need to be killed because if they're bad and going out of control, the cell should kill itself. The cell should kill cells. But so you are that guy right there. So I'll leave it up to you. Once you play the game once or twice, then you can come back here and answer the questions. And so that's all you have to do for the cell game. There are 4 scenarios for you to answer. Notice the answer choices are in the computer screens, left or right, and you have to circle it, draw an arrow to it. You can rewrite it if you want, and then that's it. So this is due Monday night for you guys, the night before our next class. And you can start playing the cell game, or if you want to keep going with the answers where we left off right here in question number 7. But let's head over to the library's rotunda If you're going to take a quick bathroom break before you get over there, that's the time to do that. Garrett, where are you? This came for you? Yeah. Garrett? James, Garrett, not here? All right, head over there. Sione's absent too. All right, see you over there. Yep, bring everything.